Good morning students. This is 11th standard botany video class, video 3. In this video, we are going to continue the lesson respiration. Dear students, we are going to see the another cycle or a stage, Krebs cycle or citric acid cycle or Tca cycle. Here, two molecules of acetyl coenzyme which is formed in the link reaction now enter into the Krebs cycle. The cycle was after named the scientist who discovered this process, that is Sir Adolph Krebs, in the year 1937. For this process, several enzymes are required. Those necessary enzymes are present in the mitochondrial matrix except succinate dehydrogenase enzyme, which is found in the mitochondrial inner membrane. So, this Krebs cycle is taken place in the mitochondrial matrix is the important 1 mark question. Dear students, see the diagram. This is the structure of mitochondria. It's a very important. Draw the following diagram in 3 marks question. The shape of the mitochondria is saucer shape, surrounded by two membrane, outer membrane and inner membrane. Outer membrane is very smooth, but inner membrane which are look like a folding like structure. The space between the two membrane is known as intermembrane space. So these two membranes are enclosed by a ground substance or a matrix. In that matrix, mitochondria contains DNA and ribosome. Then the inner membrane, they are having the finger-like projection is called cristae. It is junction. See the diagram, they are given finger-like projection. In that cristae, they are having the important respiratory substances is known as F0 and F1 particle or complexes, is otherwise known as oxysome. The structure of oxysome is look like this. It has a head region and the base. Head region is contained F1 and the base which containing the F0. Both of them are connected by a stem-like portion is called stalk. So this is the structure of mitochondria in which the Krebs cycle is taken place, particularly the mitochondrial matrix only contains many enzymes. So Tca cycle taken place in the mitochondrial matrix. See the picture. So this was the scientist Sir Adolph Krebs, was born in Germany on 25th August 1900. He was awarded for Nobel Prize for his discovery of citric acid cycle in physiology in 1953. So this Tca cycle, which is start with the process known as condensation. That means the first product of the acetyl coenzyme A, which is get reduced into citric acid with the help of the oxaloacetate in the presence of one molecule of water. It produces the citrate or citric acid. So the first compound which is formed in the Krebs cycle is known as formation of citric acid or citrate. So in this cycle, first which is formed citrate or citric acid, so that this cycle is known as citric acid cycle, that is CAC or Tca cycle. That means during the 10 steps of this series of reaction, 3 molecules of carbon dioxide is liberated outside, so that it is known as tricarboxylic acid cycle. So different enzymes which is present in the mitochondrial matrix. So different series of reaction is taken place in a cyclic manner. So during the conversion of succinyl coenzyme A to succinate by the enzyme succinyl coenzyme A synthetase, A molecule of ATP synthesis from substrate without entering the electron transport chain is known as substrate level phosphorylation. Is the important 2-mark question. That means without using the electrons, the adenosine diphosphate combined with inorganic phosphate to produce one molecule of ATP between succinic acid and succinyl coenzyme. One molecule of ATP is produced without utilizing electrons. That level of phosphorylation is known as substrate level phosphorylation. It is taken place in the plants, but in animals, a molecule of GTP, guanine triphosphate, is synthesized from guanine diphosphate with inorganic phosphate. In a couple of reactions, that means in 2 reactions, GTP is converted into GTP with simultaneous synthesis of ATP from ADP into inorganic phosphate. And also in 3 stages, that is 4th stage, 6th and 10th stage, a coenzyme NAD is get reduced into NADH2. And the 8th step, another coenzyme FAD is get reduced into FADH2. So these are all the different process which is taken place in series of reaction of Krebs cycle. Dear students, see the diagram. It's a very important 5-mark question. Draw a flow chart of Tca cycle, citric acid cycle or Krebs cycle, which contains 10 series of reaction. Before that, we are going to see the link reaction, that is oxidative decarboxylation of pyruvic acid, which is taken place in the mitochondrial membrane, that is inner membrane. A pyruvic acid or pyruvate, which is 3-carbon compound, which is produced in the glycolysis, which is converted into or condensed into acetyl coenzyme A with the help of the enzyme pyruvate dehydrogenase. One molecule of carbon dioxide is liberated. This is the first carbon dioxide which is released in this process. And also one of the coenzyme NAD is get reduced into NADH2, but this is not included in this process. So the 2-carbon compound acetyl coenzyme A, by utilizing one molecule of water and get reduced into citric acid, which is 6-carbon compound, by the process called condensation. It's given at the side, process 1 condensation, by the enzyme citrate synthase. Then the citrate, by releasing one molecule of water outside, in the presence of enzyme aconitase, converted into cis-aconitate, which is 6-carbon compound. So removal of water is known as dehydration process, given at the side, step 2. Then the step 3, cis-aconitate converted into isocitrate by utilizing one molecule of water in the presence of enzyme aconitase. So that is said to be rehydration process. Once again, re means what is that one? Once again, the water molecules enter into the reaction. Then the 4th step, isocitrate converted into oxalosuccinate, which is 6-carbon compound. At the time, one molecule of NAD get reduced into NADH2, that is oxidation process is taken place. Isn't it? Addition of hydrogen and removal of oxygen. Isn't it oxidation? So you already studied in the 4th steps, 6th step and the 10th step. the NADs get reduced into NADH2. So this process of isocitrate and oxalosuccinate taken place in the presence of enzyme isocitrate dehydrogenase. Next, this oxalosuccinate converted into alpha ketoglutarate, which is 5-carbon compound, which releasing one molecule of carbon dioxide, that is decarboxylation, is the second process of decarboxylation. Already first one is taken place between pyruvic acid and acetyl coenzyme. The another one, it is taken place between oxalosuccinate and alpha ketoglutarate. Next one, alpha ketoglutarate converted into succinyl coenzyme A by absorbing one molecule of coenzyme A or combined with coenzyme A, releasing one molecule of carbon dioxide, so decarboxylation process. The coenzyme NADs get reduced into NADH2, that is the oxidation process. So in the sixth step, two processes are taken place, oxidation and decarboxylation. In the presence of enzyme alpha ketoglutarate dehydrogenase. Then the newly formed succinyl coenzyme is a 4-carbon compound. By absorbing one molecule of water, so hydration process taken place in the seventh step, which releasing the coenzyme A outside and converted into succinate 4-carbon compound in the presence of enzyme succinyl coenzyme A synthetase. At the time, without utilizing electrons, one molecule of adenosine diphosphate is converted into ATP by adding with inorganic phosphate. So this phosphorylation is known as substrate level phosphorylation between succinate and succinyl coenzyme A. So the next one, succinate, which is 4-carbon compound converted into fumarate. This is also 4-carbon compound by the enzyme succinate dehydrogenase. Oxidation process is taken place, that is FAD get reduced into FADH2. So this one also we have seen in the paragraph. Next, the fumarate, which is absorbing one molecule of water by the enzyme fumarase, converted into another 4-carbon compound malate. Finally, the oxidation is taken place in the 10th step, that is malate converted into oxaloacetate, which is 4-carbon compound in the presence of malate dehydrogenase. The coenzyme NADs get reduced into NADH2. We already seen the NADs get reduced. This oxidation process is taken place 4th step, 6th step and also 10th step. In 8th step, the another coenzyme FADs get reduced. There is the another oxidation process. One ATP molecule is produced in the 7th step between succinate and coenzyme, succinyl coenzyme A, without utilizing electron transport. So that phosphorylation is known as substrate level phosphorylation. So this is the series of reaction which is taken place in Krebs cycle. It's very important 5-mark question which is taken place in the mitochondrial matrix. Dear students, the total link reaction and Krebs cycle in mitochondria is given here. Pyruvic acid plus 4-NAD, that is a coenzyme. The another coenzyme FAD, utilizing 4 molecule of water, adenosine diphosphate plus inorganic phosphate, which is taken place in the mitochondrial matrix, is the reactant. Giving the product is 3 molecules of carbon dioxide liberated outside, 4 NADH2 is formed, 1 FADH2 is formed, 1 water molecule is liberated outside and produce the 1 molecule of ATP. So this is the overall reaction of Krebs cycle and a link reaction which is taken place in the mitochondrial inner membrane and mitochondrial matrix. See the paragraph. 2 molecules of pyruvic acid formed at the end of glycolysis process enter into the mitochondrial matrix. So, the Krebs cycle is repeated 2 times for every glucose molecules because 2 molecules of pyruvic acid enter into the Krebs cycle. So, first 1 molecule of pyruvic acid is converted into oxaloacetate. Again, the another reaction is taken place of another pyruvic acid. So, that's why they are given Krebs cycle is repeated twice. That means 2 times. Where 2 molecules of pyruvic acid produces 6 molecules of carbon dioxide. So 2 pyruvic acid about 2. How many carbon dioxide? 3. 3 2s are what is that one? 6 molecule of carbon dioxide and 8 molecule of NADH2. So NADH2 molecule means 1 molecule means which contains 2. Isn't it? So first one cycle. See they given what is that? 4 NADH2 formed. Yeah? So 2 steps is taken place. 4 into 2, what is that one? 8 NADH2 molecules are produced. Likewise, FAD produced in 2 steps, not 2 steps, 2 reaction. First reaction, second reaction. So, what is that one? 2 molecules of FADH2 and also 2 ATP formed in 2 reaction. So, 2 molecules of ATP. So, this is the overall summary of link reaction and Krebs cycle which has taken place in mitochondria. Dear students, next we are going to see the significance of Krebs cycle is the important 3 mark and 5 mark question. This Tca cycle provide energy in the form of ATP for the metabolic process taken place in plants. And also it provides carbon skeleton, that is 3 carbon dioxide molecule is liberated, or the raw material for building up process, that is anabolic process. Third one, many intermediates of Tca cycle are metabolized, produce different compound, amino acid, protein and nucleic acid. The coenzyme which is known as succinyl coenzyme is a raw material produced in the Krebs cycle used to form the chlorophyll, cytochrome, phytochrome and other pyrrole substances. The another one, the alpha ketoglutarate which is 5 carbon compound. Another one, a 4 carbon compound oxaloacetic acid or acetate which are undergo reduction process and amination process produce amino acid. This Krebs cycle acts as a metabolic sink which is play a central role in intermediary metabolism. So these are all the different advantages of or significance of Krebs cycle is an important 5 mark question and 3 mark question. Dear students, the another important process is known as amphibolic nature. The Krebs cycle is otherwise known as Amphibolic Pathway because the 2 processes are taken place in the Krebs cycle. That is anabolism. Now only we have seen in the significance. It will be responsible for anabolic process. And energy is also produced. Catabolism is also taken place. So, this reaction is known as amphibolic pathway or nature. See the answer. Krebs cycle is primarily a catabolic pathway, breaking down of food material and gives rise to energy, which is helping to the different biosynthetic pathway, and also by an anabolic process taken place, that is building up process, is called a amphibolic pathway. So in this pathway, oxidation of carbohydrate, fats and protein is taken place. In this process, fat is act as a respiratory substrate, which is first broken into glycerol and fatty acid. Then the glycerol, which is converted into dihydroxyacetone phosphate and acetyl coenzyme A, which is entered into the Krebs cycle. So this will be produced by the broken down of fatty acid into glycerol and fats converted into glycerol and fatty acid. So fat is one of the respiratory substrate. In another one, the protein is act as a respiratory substrate, which is degraded into amino acid or producing amino acid by the enzyme proteases. So this amino acid, after deamination, enter into the Krebs cycle with the help of pyruvic acid or acetyl coenzyme A. So the respiratory intermediates form the link between synthesis as well as the breakdown. That means synthesis means anabolic process, producing. Breakdown means catabolic process. So the citric acid is the common pathway for oxidation of fuel molecules like amino acid, fatty acid and carbohydrates, that is protein, fat and carbohydrate. Therefore, this Krebs cycle respiratory pathway is known as Amphibolic Pathway. That means two processes are taken place. Breaking down of food material and also building up of the food material is taken place in the Krebs cycle. So it is known as Amphibolic Pathway. Is a very important 3 mark and 5 mark answer. Why Krebs cycle is known as Amphibolic Pathway? Dear students, I hope you have all understood Krebs cycle and Amphibolic Pathway and its significance. Up to this, I conclude my video. Remaining, we will see in the next video class. God bless you. All the best, children.